We begin the topic of cellular respiration making a statement about what this process is all about. And it is precisely about extracting energy from the food we eat, energy that will be used by cells to perform whatever activity they need to perform. And in my opening slide, I have a panda bear. It's an extraordinary animal in many ways. Hopefully, when we get to Bioto 12, we'll see more about these bears, which are indeed bears. They are not connected to or related to raccoons. The panda bear spends about 12 hours a day. Can you believe it? 12 hours a day eating. And one of the reasons they spend so much time eating is that unfortunately, bamboo, which is their favorite item in their diets, doesn't provide with as much energy for their cells as you would get from something like nuts or meat or other types of food items. And so the animal not only does it spend a lot of time eating, but also spends a lot of time sleeping. And it kind of has to do with the low energetic value of the food they eat. But we eat, and what we're hoping to get from the food we eat is going to be energy, of course, other nutrients that provide essential materials for the health of your body, tissues, cells. But let's take a look at what this class is going to help you understand. From the beginning, the first objective you will see is going to be explaining the purpose of cellular respiration, and that is exactly what I want to do in this video presentation. The word aerobic respiration is going to imply the extracting of energy from food in the presence of oxygen. And I want you to have a distinction in your minds about what aerobic respiration is and just cellular respiration. So cellular respiration involves all of the metabolic pathways for extracting energy from food, which at times can be in the presence of oxygen, like in the case of aerobic respiration, and sometimes energy can also be extracted from food in the absence of oxygen, and we're going to call that anaerobic respiration. But let's focus for a moment on aerobic respiration. There's a connection with breathing, and in fact, the word respiration sometimes can have a dual use in the English language, because respiration sometimes refers to the breathing you do, the extracting of oxygen from the surrounding air in the atmosphere so your cells can use it. And to avoid this confusion, this dichotomy, we are going to be referring to gas exchange using the word breathing, and we're going to use respiration in relationship to the extraction of energy from food. Notice, however, that the two concepts are going to be related. In the case of aerobic respiration, breathing provides the oxygen that is necessary for the maximum extraction of energy from the food we eat. When you compare aerobic respiration to anaerobic respiration, with oxygen, aerobic respiration can extract sometimes up to 20 times more energy from the food you eat compared to anaerobic respiration. And we will be seeing ATP totals as we get into this topic, the details of the topic. What I want to do in this initial video is I want to explain to you an overview of what aerobic respiration attempts to accomplish. Keep in mind that the numbers are going to vary from cell to cell, and depending on the circumstances of a cell in an organism, how much ATP can be extracted. In the explanations you will see in my class, you will see that a maximum of 38 ATP molecules can be obtained from one molecule of glucose. That maximum number, however, is rarely achieved. In most cases, cells will be able to extract from a molecule of glucose around 30 to 32 ATP molecules if oxygen is available. So here we have an equation that is going to be really important for all of you to know, to remember, but more importantly, to understand. In this overall equation for cell respiration, aerobic respiration, to be more precise, one molecule of glucose is going to interact with 6 molecules of oxygen, and it's going to yield 6 molecules of carbon dioxide and 6 molecules of water. Along the way, energy is going to be extracted, which is going to be briefly stored in adenosine triphosphate, which we also call ATP. Other forms of energy, including heat, are also going to be released as the process of aerobic respiration proceeds. So, keeping in mind the totals, please understand up to 38 ATP can be extracted. This is usually the case. Most of the time, cells cannot get that many ATP, but about 30 to 32 ATP can be extracted per molecule of glucose. So, what exactly is the energy that will be extracted from food? A molecule like glucose serves only as an example here. Other molecules can be used as a source of energy, including lipids, proteins, and even a little bit of some of the components in an in a DNA molecule. But the energy is going to be in the potential energy of electrons depending on what orbital they're occupying in the food molecule that is going to be used for metabolism. So here's what I'm saying. In a molecule of glucose, there are going to be electrons with a high level of potential energy, and those electrons will provide the energy that is needed for making ATP. What about oxygen? The role of oxygen is different. Oxygen is going to be there mostly for accepting the electrons that have been used in the process of making ATP, capture them at the end of aerobic respiration, and helping do something else with them, which in this case is going to be turning them into water. Although water is seen in this equation as a byproduct, something that is not part of the ultimate energy-rich ATP molecule, this water is important in an organism and it serves as a form of metabolic water for other activities a cell must perform. So it can be called a byproduct because it's not a source of energy, but at the same time, this is going to be a material that is being recycled and reused in the cell. The carbon dioxide that is produced is something that cells must dispose of. Cells must release it, and we do it whenever we exhale. During breathing, gas exchange, we keep oxygen, and we exhale the CO2. Cells have also produced as a byproduct during aerobic respiration. Because there is going to be a transfer of electrons. Glucose is giving off electrons, and oxygen is capturing electrons. From a chemistry point of view, this is going to be an example of a redox reaction. Reduction is going to be a short for reduction and oxidation. Reduction in chemistry is going to be when a substance gains electrons and hydrogen ions. Oxidation is going to be when a substance loses electrons and hydrogen ions. And here you have to be mindful that when you think about reduction in everyday life, you think about something getting smaller or you're getting less of something. Here in this case, reduction means a lowering of the negativity of a molecule, but in reality, reduction means that something is gaining. It's gaining electrons and hydrogen ions. Let's apply these two concepts to this summary of the equation for cell respiration we have for aerobic respiration. So glucose, in this case, is going to be losing electrons, and because glucose is losing electrons, it's being oxidized. It's going to be when a substance loses electrons and hydrogen ions. And think what happens to glucose if you remove all of these hydrogens you see here in the middle. All you would have left is carbon and oxygen. Well, something you can make with carbon and oxygen is going to be exactly carbon dioxide. What happened to the electrons and hydrogen ions that were stripped from glucose? Oxygen is going to pick them up. And what can we make if we added hydrogen and electrons to oxygen? Hydrogen and oxygen can make something like water. Because oxygen is gaining electrons and hydrogen ions, oxygen is being reduced. And that is what you see here indicated in this equation. Oxygen is reduced to form water. So these are some concepts that you need to spend some time thinking about it. What is the meaning of redox? What is the meaning of reduction and oxidation, and how it applies in the process of aerobic respiration? Because electrons have to be removed, and this is a real precise type of a reaction, we need enzymes. By now you've learned that enzymes can catalyze reactions in the cell, meaning can make the reactions happen faster. And that is a way in which enzymes can bring substrates together or allow for a transformation in the substrate so that it can produce something the cell desires. In this case, enzymes called dehydrogenases are in charge of removing hydrogen ions and electrons. In a sense, hydrogen atoms are being removed from glucose molecules or from any other fuel molecule that is being used in metabolism. I like the name of this enzyme, dehydrogenase. The first part, D, means to undo or to remove. Hydrogen, well, we know that's the first element in the periodic table, atomic number 1. It has 1 proton and 1 electron. So the enzyme dehydrogenase is removing hydrogens from fuel molecules. This enzyme dehydrogenase is going to allow for something to pick up the electrons, and that something is going to be like an electron shuttle. Imagine it like a cab, like an Uber that is going to be transporting not people, not passengers, but this special Uber or Lyft we have inside of the cell is going to be transporting passengers called electrons. Electrons are going to travel in pairs. Each molecule of NAD, which is like a coenzyme, can carry 2 electrons. And you can see what happens. I want to see if I can expand on this illustration that I have. Look at the work of dehydrogenase. On this part of the illustration, we see a portion of a fuel molecule like glucose. The gray dots indicate carbons. There you see the hydrogens, the oxygens. So the enzyme dehydrogenase is going to catalyze an oxidation reaction, the losing of electrons by this fuel molecule. And what is going to left behind is going to be those carbons in the carbon skeleton, a single oxygen. These 2 hydrogens you see on the left are now hydrogen atoms that have been removed by the action of dehydrogenase. And what is going to happen next? These hydrogen atoms are going to be picked up by NAD+. Remember, each hydrogen has 1 electron and 1 proton or hydrogen ion. What NAD+, nicotinamide adenine dinucleotide, picks up is going to be precisely 2 electrons. The electrons can be seen here as blue dots. So there we have 2 electrons. NAD+ is also going to pick up 1 hydrogen because this hydrogen ion is positive, and you have another positive charge here that combines with the 2 negatives of the 2 electrons, and it produces an electrically neutral electron shuttle or electron carrier that is going to provide electrons for the process in cell respiration where electrons are needed to make ATP. So that's the role of these coenzymes, NAD+ and FAD. They are like shuttles. They are like vehicles that are transporting passengers, and passengers in this case are going to be electrons. One more thing I want you to notice about this illustration is that there are 2 forms of the coenzyme. The one on the left, NAD+, is the oxidized version of the coenzyme. The one on the right, NAD+, is going to be the reduced version because it has gained electrons and a hydrogen ion. 1 hydrogen ion is left alone, and it will participate also later in the process of making ATP. So what happens to these electrons? The electrons are going to be delivered to inside of the mitochondrion, where along the inner mitochondrial membrane, mitochondrion has two membranes, one on the outside, one on the inside with many folds. Along those folds, there are these protein complexes of the electron transport chain. And as electrons go from one protein complex to the next one in the electron transport chain, small bits of energy are being given off that will be eventually used for the making of the most ATP. The electron transport chain and chemiosmosis is the stage of aerobic respiration that produces the most ATP. Notice how here at the end is where oxygen will be needed to pick up a pair of electrons, also one hydrogen ion, and two hydrogen ions, and form water, H2O. Here in this illustration, I have two of those fuel molecules your cells can use at any moment. Glucose, the preferred fuel molecule by all forms of life, from bacteria to the most complex plants and animals, they like to use glucose. Fructose is also a monosaccharide. And you can see here all of these hydrogens. Every hydrogen you see in this molecule of fructose, every hydrogen in the molecule of glucose can provide an electron that will be used in the synthesis of ATP. So aerobic respiration is going to require three main stages. The first one, glycolysis, which simply means the splitting of a sugar. Glyco means sugar, lysis means splitting or cutting. This is going to be happening in the cytoplasm of the cell, outside of the mitochondrion. And if you're thinking, why is it saying the word funny? Mitochondrion is one organ and mitochondria is many. And so before you try to correct me, if I'm using the word mitochondrion, I'm referring to one of these organelles. Okay, let's continue. So next, glycolysis will produce something called pyruvate, which is also considered a carbohydrate, a sugar, and it will be shipped to the inside of the mitochondrion. You see the word right here. And that is where the beginning of the citric acid cycle will take place. Before the citric acid cycle begins, there is a preparatory stage called the grooming of pyruvate or pyruvate oxidation. There are two ways you can refer to this. And then the citric acid cycle takes place inside of the mitochondrial matrix. The matrix is this thick, jelly-like fluid inside of the mitochondrion. And from here, electrons that have been extracted from glucose in glycolysis, more electrons extracted from pyruvate and acetyl-CoA, now those electrons are traveling in NADH and FADH2. Those electrons will be delivered to the final stage of aerobic respiration, which is called oxidative phosphorylation, and that takes place along those protein complexes I described before in the inner mitochondrial membrane. There are more videos I will be providing for you to look and visualize how these three stages of cell respiration, aerobic respiration, contribute to the making of ATP for a cell in the presence of oxygen.